little over 2.5 years now. He is a UMass Dartmouth alum with undergrad, Fall River before that with Durfee. And he was one of those other students, many other students that I harassed into coming and doing immunology. I don't think he could ever possibly say he thought his research to do a master's would have been in immunology, let alone fish. But Greg joined my lab. He has been just a fantastic lab member to actually have in my lab. He has worked incredibly well with everybody and been part of a really good team. And I am going to be a lot sad to actually lose him from there, because he is now one of my most productive members in my lab, and we were just talking about this and then they leave. But Greg's work today that he's actually going to present is essentially the culmination of his master's thesis. And just recently, actually a few days ago, he had his work actually accepted for publication. Essentially no changes now, which is a really great accomplishment to actually have that done before it comes out. But even more to the point, last week when I was down in DC, one of the reviewers of the paper came up and spoke to me and just said that Greg's work has really put together what the field hasn't had for about 20 years. So I was really chuffed to hear that myself, but there's a testament to Greg. Greg did actually add more to this project than what I could have done myself, and especially from his ecology and background from undergrad, really actually supported the writing and the way in which he thought about the topic that he was going on. So today, you better live up to that now, Greg. He will be presenting his master's thesis defense.
Great. Good afternoon, everybody. I had to compete with a royal wedding in this great weather, so I'm glad for the turnout. Today, I'm going to be presenting on IgM dynamics in fish and how that changes throughout the immune response. Now, for you, the consumer, what this means is a cheaper fillet of fish. And hopefully, by the end of the talk, you know what I mean by this. So let's get into aquaculture. Aquaculture is fish farming, and it's actually the fastest growing food sector. It currently provides 50% of our seafood, as opposed to the wild caught fisheries, which haven't been growing since the 1970s. Now, here we have a small fish farm in Massachusetts where we actually obtain our rainbow trout, and it's been in operation since the 1800s. Now, ironically, it's the very intensification of fish farming that's challenging its growth. Now, what do I mean by that? Well, with the intensification of fish farming, you're getting more fish huddled together, so they're likely to transmit diseases, and you also get transmission of pathogens from one location to another. These outbreaks of diseases are known as epizootics. Now, bacteria are largely responsible for these epizootics, but also fungi, viruses, parasites can all lead to outbreaks and diseases. Now, in 2002, infectious salmon anemia was responsible for a 24% loss of annual production in Atlantic salmon in New Brunswick, Canada. In 2009, when we looked at rainbow trout, all the mortalities, so it could be due to changes in temperature, changes in salinity, could be due to handling the fish, 90% of those losses were due to disease. So to combat the diseases, traditionally, chemotherapeutics and antibiotics have been used, but they have disadvantages. So malachite green here is great at fighting off parasites, but it's been demonstrated to be a carcinogen. That is, it causes cancer in humans. In antibiotics, as we all know, its overuse leads to resistant bacteria. So in aquaculture, we now have antibiotic-resistant bacteria. This is where vaccines come into play. And since 1976, vaccines have been used for use in aquaculture, and they have advantage of taking advantage of the fish's immune system to generate antibodies. And this generation of antibodies for protection is known as humoral immunity. However, since 1976, the USDA has only licensed 13 vaccines for use in aquaculture. And many of these vaccines are for the same pathogen. So for example, 4 alone are just for Vibrio. So why do we have so few vaccines? Well, as was said, as far back as 1975, Until we truly understand the immune system of fish, can we develop efficacious vaccines. And that's hopefully where my research comes into play, where we understand the fish immune system so we can develop better vaccines, which for you, the consumer, means greater production of fish and cheaper fish. Now, how does vaccination work? Well, here I am vaccinating one of my fish, and what I'm doing is introducing Streptococcus ineae, a killed protein. And what I'm introducing is an antigen. An antigen is short for antibody generator. It's anything that stimulates the immune system. So here we have a B cell, and here's its receptor, and it's going to bind the antigen. It's then going to be stimulated. It's going to be stimulated to divide, and it's going to produce antibody-secreting cells, which are also B cells. And these are going to secrete antibody to bind the antigens. And these antibodies are essentially secreted receptors. Now, how do antibodies work? Well, they work through different mechanisms, and these are known as effector functions. There are many different types of effector functions, but I've shown 3 here. Neutralization, where you bind the antigen and prevent it from harming the host. Opsonization, where you coat it and you make it tasty for the immune system, essentially. Macrophages want to come along and ingest it. And then finally, complement activation, which results in a pathway that ultimately leads to the destruction of the pathogen. Now, in mammals, you have many different antibodies, and these are specialized for these different effector functions. So IgA here is great at neutralization. IgG is great at opsonization. IgM is great at complement activation. Now, as I said, each of these isotypes is specialized for a certain effector function. And also, they vary in the location of the body and also the amount that they're present in. So, for example, IgA is almost solely present in the mucus. Now, in contrast, fish rely essentially on a single molecule to carry out all the effector functions that mammals do. And in contrast to the mammals, it's a tetrameric molecule, meaning it has 4 arms, versus the mammalian, which is a pentameric molecule. It has 5 arms. But one thing that these antibodies all have in common is this generalized structure. As you can see, it's divided in essentially 2 discrete units. The antibody fragment, which contains an antigen binding site, and it binds the antigen with what has been described as a lock and key fit. That is, you have one antibody per epitope. An epitope is the region of the antigen to which it binds. And then you have the constant fragment, which is this tail region here. And that's what carries out the effector functions that I was mentioning. So for example, opsonization, complement activation. So how do we define a successful vaccine? Well, there are many different criteria. The most obvious one is the quantity of the antibody that you produce following vaccination. And this quantity is what we call titer in immunology. And in fish, titers to different pathogens such as parasites like ick, bacteria such as Vibrio, and viruses like viral hemorrhagic septicemia have shown to protect the fish from infection. However, fish do not develop the same quantity of antibodies as mammals. That is, they don't develop the same titer. So if a fish is infected with a pathogen the second time in its life, it will only develop titers that are about 10 times the starting level. In contrast, mammals develop titers that are thousands of times the starting level. Also, the specificity of the antibody is crucial for protection. So it may be more important that low titers are generated to the correct epitope, that is the correct binding site, than having high titers to the incorrect epitopes. And just to put this in perspective, something you'll understand, let's say you're infected with influenza, the flu. Your body is going to generate antibodies to many different antigens. For example, hemagglutinin, you have neuraminidase, difficult to say. But for the antibody to be effective, it needs to bind this antigen, hemagglutinin. And specifically, the epitope that it needs to bind to is the stalk here. Now, most of the antibodies that your body generates are going to be ineffectual to the flu. That's where vaccines come along. You want vaccines to generate them to the stalk. And this principle is very similar to VHS in fish, where the antibody needs to bind to the G protein, not irrelevant proteins such as the M protein, because the G protein is involved in penetration of the virus into cells. Now, also a hallmark of immunity is the generation of high affinity antibodies throughout the immune response. This is known as affinity maturation. And it's very similar to Darwinian fitness. So let's picture two B cells here. One B cell is going to bind antigen with high affinity. One B cell is going to bind with low affinity. So what's going to happen when that antigen concentration drops? The high affinity B cell is going to win. Thus, it's going to be the one that survives, produces antibody. And thus we get a transition of low affinity antibody throughout the immune response to high affinity as it progresses. Now, in mammals tend to class switch their antibody. So they'll go from low affinity antibody that's generally characterized by IgM to IgG, which is a high affinity antibody. But all antibodies can go through affinity maturation. Now, what in fish, it's unknown if they actually undergo affinity maturation. There's really been no proof if they do. And I was talking about class switching. Fish do not class switch. That is, they don't have IgG, IgA, all those different antibodies. They rely primarily on IgM. Now, as you remember from a few weeks ago, they do have additional antibodies, but 95% of their antibody is this IgM. Lastly, and possibly most importantly, we want vaccines to generate immunological memory. And there are two forms of memory. You can have the generation of memory B cells, that's a classical form of memory, and you can have long-lived plasma cells. Now, as mammals, we tend to rely on memory B cells. And just think, if you're infected with chickenpox early in your life, chances are you're not going to get chickenpox again. And this is due to the presence of memory B cells. Now, fish do not have memory B cells. So I found this quote online. The fish immune system is simple relative to mammals. And let's see if that's the case. So following vaccination, fish do generate titers, but it's not to the same magnitude of mammals. They do experience an increase in the specificity of their antibody that will bind to different proteins throughout the immune response. However, they do not class switch. And as I said, they rely predominantly on IgM. It's really unknown if they undergo affinity maturation, that is, that increase in affinity of antibody. And also, they don't have that classical form of memory, the memory B cells. So it's not surprising that for the past 30 years, vaccinologists have strictly focused predominantly on titer following vaccination to assess their vaccines and the specificity to which the antibodies bind. But let's think of what this is based on. It's based on the mammalian system. And what do we know? Fish aren't mammals. Thus, we have to look at fish as unique organisms. And just to give an example, here I'm pointing out the anterior kidney. And that's unique to fish because rather than having bone marrow, this is the predominant organ they rely on. Now, within that immune organ, they possess long-lived plasma cells that are secreting antibody throughout the lifetime of the fish. So their form of memory is known as humoral memory. They don't rely on the classical form of memory that I was discussing. Now, if we view fish as unique organisms, we may see that they have alternative means of fighting pathogens from mammals. And what I want you to notice is, even though I said fish rely on that single IgM predominantly, IgM is actually a very dynamic molecule in fish. So what we have here in red are disulfide bonds. And what you can see is the bonding can go from fully cross-linked, intermediate, one, it can actually go to no cross-linking at all. And just think of how this is going to affect the rigidity of the antibody. If it's fully bound, it's going to be like tying your hand with twine. It's going to be rigid. No bond, it's going to be flexible. But what I want you to realize is even though you don't see bonds here, this is always fully tetramized. That is, they're always bound together, even if it's with weak bonds. So this arm isn't just